of my favorite subjects, which is the ecology of lampreys. The talk that I prepared for you really has three parts. First, I want to talk about what are lampreys and why are they so interesting. Secondly, I want to review for you the history of the sea lamprey invasion of the upper Great Lakes and why it was such an ecological disaster. And then thirdly, I'm going to talk to you about some of the research that I and my graduate students at Michigan Tech did on the ecology of lamprey larvae in streams. They're just sort of a fascinating group of organisms from an ecological point of view, but what we learn about them may at some point be helpful in managing the impact of the sea lamprey invasion as well. So first, let's talk about what lampreys are. Lampreys are extremely ancient and primitive fish. On your right there is an evolutionary tree. And the most primitive organisms are at the bottom, and it goes up to the most derived or advanced. So these are the chordates, animals that have a notochord, and that's the core typically of the backbone. And the evolutionary time moves from left to right across the bottom here and from bottom up. There are two groups of chordates here, the lampreys and the hagfishes, that are the only surviving individuals from the late Devonian period. There were thousands of fish species in the late Devonian, but only these few have survived the last 360 plus million years. So these are survivors. Okay? They have lasted a very long time. They're so primitive that they evolved so early in the evolution of vertebrate animals that they evolved before the development of the kinds of jaws that are typical of vertebrate animals like ourselves. So they don't have jaws. They have an oral disc rather than a jaw. They're inordinately successful in an evolutionary sense because they've survived 4 major extinction events in which much of the fauna on the planet Earth was wiped out by something like the meteorite strike that supposedly, hypothetically wiped out the dinosaurs and things like that. So over the last 360 million years, there have been 4 major extinction events, and they've survived them all. So these creatures have got something going. Okay? It's working well. This is a diagram showing the life cycle of a typical lamprey. This diagram is designed specifically for sea lampreys in the Great Lakes. It's one developed by the Great Lakes Fishery Commission, but the general features are typical of all lampreys. So we can begin looking at it here in March and July when the adults spawn. They leave the Great Lakes, they swim up streams, and they swim up pretty far into the stream until they find gravel beds. They lay their eggs in the gravel beds. One female can produce about 100,000 eggs. The eggs rest for about 2 weeks and develop slowly in the gravel beds, and then the larvae emerge. When they emerge, they are only a few millimeters in length. They drift downstream with the current, and then when they get to areas of soft sediment, they burrow into the sediment and make these little burrows where they live. This larval phase here ranges from 3 to more than 10 years, so it's pretty variable. After they have grown enough to get up to, for sea lampreys, it's about 9 or 10 inches in length. Then they go through a process of metamorphosis. The larvae here have an undifferentiated mouth that's referred to as an oral hood. It's just sort of a flap of skin. And they don't have eyes. When they metamorphose into the juvenile form, they develop eyes, and they develop an oral disc here that they use for feeding. They leave the streams anytime from about June through March and drift downstream into the Great Lakes and become, in the case of sea lampreys, become predators on the larger fish species in the lake. So this is a typical life cycle.
Steve, are they making little nest there with their mouth?
Yes.
Sucking on rocks?
Yeah, they move the rocks around to make a little nest in which they lay the eggs. And the genus of the sea lamprey is Petromyzon, which in the Greek means stone sucker. So they were observed, sucking on the stones to move them around as they're doing it. So this is a typical life cycle. There are now 38 living species of lampreys spread across the temperate waters of the world. Of them, 18 species have juveniles that are predatory, and 20 have juveniles that are non-predatory. And interestingly, they occur typically in pairs. So in the streams that flow into Lake Michigan and the streams that we had in the UP, you would commonly find both the chestnut lamprey and the northern brook lamprey together. The chestnut lamprey has predatory juveniles. You can see these sharp little pointy teeth they have here for attacking their prey. But the northern brook lamprey doesn't have those teeth. And in fact, the juveniles and adults have a very short lifespan in which they don't feed, they just reproduce and then die. So it's interesting to speculate how the balance is developed between the predatory and the non-predatory species being together in so many different streams. The chestnut lamprey and northern brook lamprey, when they are mature as adults, are a maximum of about 7 inches long. The sea lampreys are not native to the upper Great Lakes. They were prevented from getting into the Great Lakes by Niagara Falls until about 1919, when modifications to the Welland Canal made it possible for them to swim up and get into Lake Erie. And from there they spread all throughout the Great Lakes. So over a period of about 20 years then they got through all the lakes. They are much larger than the native lampreys, so a large sea lamprey is 47 inches long, by comparison to 7 inches on the part of the endemic, naturally occurring lampreys. So the larger ones were capable of doing so much more damage to the larger fish in the Great Lakes that they caused the collapse of the major fish populations. And just to illustrate the collapse, in 7 years, the Lake Michigan fishery went from producing 5.5 million pounds of fish to producing 402 pounds. Okay. Now, this 402 probably reflects the fact that fishermen gave up. Okay. There are some fish out there, but it wasn't worth going after them because they were so few. So the sea lampreys really pulled the rug out from under the Great Lakes fishery in all of the Great Lakes. Lake Superior was the last one to be severely impacted, but it was impacted severely as well. In response to this, a treaty was developed between the U.S. and Canada to create the Great Lakes Fishery Commission. It was created in 1955, and its mandate was to control sea lamprey populations and then to rehabilitate the fisheries if they could find ways to do those things. First efforts involved trying to prevent the adults from swimming upstream where they could lay their eggs. For reasons that are not fully understood at this point, lampreys spawn in only about 10% of the available streams. And every year they home to those same streams to reproduce. So building barriers to migrating lampreys was a little more practical because you didn't have to build one in every river, you just had to find the 10% that they were using. This shows what's called a low head dam, and the idea here is that fish that you want to have spawning upstream can jump over it, but lampreys can't. Another approach that was similar to preventing the adults from getting upstream was weirs that directed the fish into cages, and then U.S. Fish and Wildlife Service personnel would come, pick up the cage, send the good fish upstream, and destroy the lampreys. Was the plan. This ultimately didn't work, and the reason it didn't work is that when there are floods, the lampreys figure out how to get around the structure. And when the cages are not fitting perfectly in the slot they get under the cage, they find their ways around. And at 100,000 eggs per female, it doesn't take too many to bypass the structure before they can be quite productive in laying eggs upstream. So the second approach was to survey the streams and monitor the development of the larvae in the stream and to see how they're growing. And when they get up to a size where it looks like they could be ready to metamorphose, then to poison them. So this is a U.S. Fish and Wildlife Service employee biologist who's using a shocking device to shock the lamprey larvae so they come up out of the sediment, and then you can use the nets on the end of those electrodes to pick them up and collect them. And this is a bunch of different ones that he's collected. These are the larvae. You can, well, it's not clear enough to see for sure, but they don't have eyes and they don't have a differentiated mouth. They're a typical larvae. None of them has begun to metamorphose yet. You can see they're different sizes. So they are growing, but they're not up to the size that means they could become adults yet. So when it looks like they're getting close to metamorphosing, a chemical, which is referred to as a lampricide, is put in the water, and it's administered very conscientiously to achieve just the right concentration over a period of several hours. And it kills the lamprey larvae. Now, this has really been the basis for the success of the rehabilitation of the Great Lakes fishery. It's very effective. It doesn't get every last larvae, but it gets about 98% or 99%. There are a few that are sort of hiding under the banks and things like that that don't reach. It's very effective. But there are three reasons that people are not so happy with it. One is it's very expensive. The chemical that's used is produced by Bayer in Germany, and it's very expensive to buy. And then they have all the personnel costs of its application. So it's an expensive way to control the lamprey populations. Secondly, just poisoning streams doesn't sound like a great plan. You know, it's not a publicly popular approach. And then thirdly, the lampricide has been developed to target lampreys, but it kills other things too. In particular, young sturgeon seem to be subject, being injured by it. And so the U.S. Fish and Wildlife Service has for years been looking to alternatives for ways to control the population. And they're very systematic and very thorough about exploring every possibility in terms of putting together a package of integrated wildlife management techniques, all of which together will suppress the lamprey population. And one of the things that was learned as a result of all the survey work being done by the U.S. Fish and Wildlife Survey agents was that when they compared different streams, they found that the rates of larval development varied from one stream to the next, and that the rate was proportional to the density of larvae in the streams. So when the larvae were abundant, they developed more slowly and they had a higher mortality rate or a lower survivorship rate. And this same relationship was tested in aquariums and found to be present there as well. So if you had 10 aquariums and you put in 10 different concentrations of lamprey larvae, the ones with the highest density of larvae grew the slowest and had the highest mortality rate. So this was an interesting observation, but it wasn't obvious why this would happen. How is it that they somehow inhibit each other? When ecologists see these kinds of density-dependent effects, one of the things that comes to mind immediately is competition, and more often than not, it would be competition for food. But what do we know about feeding of the larvae and what's important to them? We know that in the burrows where they live, they have these burrows that they build, and there's a little connection, a tube to the water above, and then they have a respiratory current in which they pump water through their gills and down into the burrow below them. And in the process, there are all these little particles that are suspended in the water, and they capture these in the mucus that is on their gills, and then that makes their diet. So we know just that much about how they feed. But we don't know whether the limiting factor is the quantity of food that's available to them or the quality of the food. It could be either, or it could be some combination of the two. And then after that, we would like to know how density affects their use of the food, if there is indeed a relationship there. And so when you examine their gut contents, this is what you see. This is a photograph taken through a microscope, a light microscope. And the first scientists to look at the diets of lampreys said, oh, well, it's clear what they're eating. There's an algal cell there, diatom, another one there, there's one, there's one. There's a couple here. So they're eating algae. Okay, that was the conclusion. Here's another example of the material that they feed on. And again, you can see an algal cell right there. And there's one there. But the early biologists made the mistake of ignoring all the rest of this stuff here. Okay? And it turns out that when you use chemical techniques to identify all the microbes that are present, they make up less than 5% of the organic matter in the gut contents. More than 95% is this amorphous material here. Now, we could take the rest of the evening and more to talk about what it is and where it comes from. So, just to give you the Cliff Notes approach, what this material turns out to be is organic matter that's secreted by microorganisms to protect them from the outside environment, which accumulates because they keep producing it. And then eventually it sloughs off and it washes down the stream as those little particles that we saw. So biologists now refer to this material as biofilm. And biofilm is everywhere. Okay? If you scrape your thumb along the inside of an aquarium, it's going to come off with a lot of slime on it. That's biofilm. If you check your teeth in the morning, scrape them with your thumb, that's biofilm. Okay? The microbes that live in your mouth have produced that protective layer that you're going to brush off. So this stuff is everywhere, and it's organic, and therefore it's potentially a valuable food resource. So we inferred that food quantity per se was not going to be a limiting factor because there's so much of this biofilm in the streams that the lampreys couldn't begin to make a dent in it. There's more than enough. If you measure the concentration of particles above a group of lampreys and below a group of lampreys in the stream, there's no measurable difference. So they're removing a minute fraction of the total material. But diet quality is another possibility. And we know from research on other animals that feed on biofilm that both the total amount of organic matter that's in the sample and the fraction of that organic matter that is protein, both of those are important. Those are macronutrients that support animal growth in the natural environment. Now, it's important how much of that is in the diet, but it's even more important how much of it they can digest. Okay? So that's a second measure direct measure of food quality. The third measure of food quality is plumpness. Okay, plumpness may sound a little silly, but in fact, if you calculate the weight per length for fishes, where this is used a lot, the larger that number is, the plumper they are. And it's given the name, the ratio is called the condition factor. And we know that animals of the same species, when you compare them, if the condition factor is higher, they have a better diet and they're in better health than if it's low. So if they're really skinny, that number would be low. If they're nice and plump, it would be higher. So we have these three measures of diet quality. But before we can actually go out and compare diet quality in the streams, we need to consider how quality varies from one location to the next in the stream before we can talk about the effects of density. The U.S. Fish and Wildlife Survey agents early in their work began to distinguish three habitat types, and they would sample them in a stratified way to develop reliable estimates of population density in streams. So type 1 is the organic matter that accumulates in streams, it's sort of soft sediment. Type 2 is sand sediment, and type 3 is gravel. Well, the lampreys larvae are never found in type 3. They don't inhabit gravel areas. They're found in sand areas, but not very often, only infrequently. They are found most abundantly in the type 1 organic sediment areas, but there's a special type of type 1 habitat where they are extra abundant, and we've given that the name of type 1A. Type 1A sediment is behind some obstacle to flow that creates a sedimentary environment, where this very fine flocculent organic matter settles. And that's where the lamprey larvae aggregate. Okay? Now, make a mental note of this because we're looking at density effects, but their own behavior causes them to aggregate and increase density in patches. Okay? Now, when we compare food quality of stomach samples taken from larvae in type 2, type 1, and type 1A habitats, this is what we learn. We're comparing here the diet and then the assimilation of the diet, which is the extent to which it's digested and then assimilated, and then finally the condition factor. It just stands for ash-free dry weight, which is a measure of organic matter. And then this is the concentration of amino acids, and it's compared for type 2, type 1, and type 1A habitats. So what we learn from this is that there's no effect of habitat type on the quantity of either organic matter or amino acids in the stomach contents. But there's a huge effect on its digestibility. So there's something qualitatively different about the biofilm that the lamprey larvae are getting in the preferred habitat. And look at the difference it has on condition factors. Okay? The ones that are in type 1A habitat are a whole lot healthier and plumper than the ones that are found in just type 1 or type 2. So what this means is if we want to understand the effects of density, everything that we are interested in is happening in type 1A habitat. That's where the lamprey larvae are aggregating. That's where they're getting the food resources they need to grow rapidly. So we wanted to see if we could identify effects of density on the actual larvae in the streams in specific patches of 1A habitat. We took two approaches. One was involving in-stream cages. So a cage consists of a plastic tray that's 3 inches deep and it's filled with type 1A sediment. And then it's covered over with a screen that prevents the lamprey that are in the cage from leaving, or it prevents other lampreys from coming in. So we can control density that way. So we arranged cages with 3 different densities, 5, 10, and 15 larvae per cage. And then we replicated each of those 3. So there were 9 cages in a given patch of 1A habitat in a stream, and we did this in 3 different streams. And we left the cages in there for a week, and then we collected the larvae and analyzed their gut contents. And this is what we found. Another measure that we wanted to check on was gut fullness here, which is an indication of how actively they're feeding. And there was no effect here. The gut fullness is shown by the open squares. There's no significant effect of density on the feeding rate. There was also no effect of density on the composition of the diet, but there was a pronounced effect on the digestion and assimilation of both organic matter, which is shown as the dark circles, and of protein, or amino acids, as shown as the open circles. So we found, in fact, what had been inferred by the survey agents we could demonstrate experimentally using cages. Cages are always a good way to produce a controlled environment, but it's not a natural environment. So we wanted to take another step to see if we could see the same thing for animals that were not being affected by the experimental conditions. And we did that with some surveys, again in 3 different rivers, and we took a large patch of type A habitat and we laid out a grid over it. And then each square in the grid was sampled, and we used that backpack shocker that you saw and collected all the larvae from that patch and saved them for analysis later on. When we did that, we found that there were definitely areas where the larvae were congregating. So there are 2 sort of hot spots here. There were 10 larvae per grid in this area that's yellow here and here, and then much of the rest of the area, there weren't that many, 2 or 1 in some cases. So that meant we had a range of densities that we could compare, which is what we were hoping for. And here are the effects. There was no effective density on either gut fullness or condition factor. Okay, so that's a little different from the experiments that we did with cages, because that did show an effect of condition factor. But there was a major effect of density on assimilation of both of the nutrients that we're interested in, total organic matter and protein. And the slope of these lines is dramatic. As you can see, it's not a subtle effect, it's really a pretty pronounced effect. So here are the conclusions then from these studies. One is that lamprey larvae aggregate in habitats that provide the high quality food. Secondly, that crowding does not affect feeding rate, but it greatly reduces assimilation efficiency. And thirdly, the benefit from higher food quality is greater than the cost of crowding, as the crowding interferes with the digestive processes. We know this because the condition factors of larvae collected from type 1A habitat are so much greater than from other habitats. So here's the final slide. The question is, how does this make sense? Because you have sort of a paradox here. You have these animals congregating in an area where they seem to interfere with each other's success. And the way we think we understand this is that because the lamprey larvae aggregate in this type 1A habitat, they're especially vulnerable to predators that would feed on them. So there are a variety of fishes that sort of dig in the sediment looking for prey. And if those predators have learned, which surely they do, that when you find like a big log in the stream and there's a lot of soft sediment behind it, that's going to be a great place to find lamprey larvae. Now, this is not good for a lamprey larvae because some of them have got 10 years to go before they can reproduce. And so with such a long larval phase, avoiding a predator has got to be a super high priority because you lose if you can't succeed at doing that. It turns out that these lamprey larvae are very, very sensitive to disturbance. We learned this when we were keeping them in an aquarium. If you take a pencil and just gently stir the sand about an inch away from them, they flee. And the way that they flee is they move horizontally through the sediment, sideways, until they hit the side of the aquarium. Then they come up in the water and swim frantically around the aquarium for a few minutes until they finally go back into the sediment. So they're very, very sensitive to disturbance. In areas where they're concentrated, the movement of one is likely to disturb the others close to it because they're often only an inch or two apart in these concentrated areas. And so the density is going to cause frequent disturbance, which you can reasonably infer would interfere with their digestion of their diet. You can think of two possible ways that might be the case. One is that it might just interfere with their feeding and pulling in the right kinds of particles that are digestible. Another is that the basic enzymatic process of digesting their diet might be interfered with. When humans are anxious, they don't digest their diet nearly as well. And the same thing is a possibility, at least for the lampreys. We don't know the biochemistry of lampreys digesting biofilm. So, to sum up then, this is how this particular group of animals has managed to persist over 360 million years. They live in an environment that's highly predictable. There's always this rain of biofilm coming down the stream. They are in balance with the rest of the system. The predatory ones can prey on other fishes in the streams or the lakes that they might enter, but they are not large enough to interfere with the stability of those prey populations. And it seems to be that the same process continuously year after year, not very subject to short-term climate variation, and so they have persisted for a long, long time. So I would be happy to answer any questions if I can.
I have a question that I hadn't thought about, even though I've talked about this in class. So the fishes in Lake Erie, Michigan, Lake Huron, and Lake Superior had never encountered sea lampreys before, and they didn't have really very many defenses against them. But the fishes that live in Lake Ontario are able to withstand the onslaught of sea lampreys because they come down the St. Lawrence River. So is there any, do the sea lampreys have any effect on the fisheries in Lake Ontario? Because they have a long history with sea lampreys.
Yeah, that's a great question. For whatever reason, sea lampreys are not abundant in Lake Ontario. The first one was reported early in the 1800s, so we know that they're there. It's been known that they're there for a long time, but they have not achieved large population sizes that have a big impact on the native fish population. And I'm sure there are people who know a lot more about that particular situation than I can help with. But not only is Lake Ontario open to invasion by sea lampreys, but a lot of other fishes from the St. Lawrence Seaway. And so it may be that some of those other fishes are predators on sea lampreys.
Right.
And sea lampreys don't get out of control in the ecological sense in the Atlantic Ocean because there's so many big fish that can eat them.
Well, this just gets the big fish and bring them into Lake Michigan.
Like salmon.
Like salmon.
They're not that big, I guess. They're sharks.
What about bird predation on larvae? Are there,
I have never seen an account of that. So I don't know if it happens.
so it's really mud scavenging fish.
Yeah. And it's not common to find lamprey larvae in the stomachs of fish. But it does happen. And the speculation is it happens usually when there's a spate that completely stirs everything up, and the lamprey get displaced from their burrows and exposed, and that's when the predators get them. Well, there's a bird connection here, and that is that this biofilm material, it turns out, is the food of several species in the genus Colibris. So those are sandpipers. Before we retired and moved to Michigan, we used to spend a fair amount of time on the Georgia coast. And the most common bird for the years that we were there was sanderling. It's Colibris alba. And it feeds by chasing the receding wave and sort of vibrating its bill in the water and collecting biofilm. And the western sandpiper is another one that's been studied pretty intensively in the Vancouver area, and it does the same thing. So these birds are feeding on biofilm and doing well at it. And it would be interesting to learn more about how selective they are. Do they select for high amino acid biofilm? Do they have any adaptations for its digestion? Biofilm is also the main food of a lot of fishes in the tropics that are super abundant. So there's one fish species that I worked on in Argentina for several years. Its common name in Argentina is the sabalo, but its scientific name is Proteolotus lineatus. You might have seen that in the literature at some point. There are 207 species of fish described from the middle Rio Paraná. One species, this one species makes up 60% of the total fish biomass. So it is like a bee fish of the river system there. Its young are the prey of all the predatory fishes, and yet it feeds on this biofilm material. And it's pretty sophisticated in the way it does it.
How does it suck it off rocks? Is that,
No, it actually sucks it off the bottom in areas where the water is rising, so that you have soils that were previously dry that then get wetted by rising river water levels, and that creates a burst of microbial activity, and they get in there at the right time when the protein content of that biofilm is at its maximum. They know how to find that. And it produces this impressive growth.
So it almost seems as if the species that can feed on biofilm, because it's so prevalent, seem to have some evolutionary advantage.
They have a huge advantage because it's essentially an inexhaustible source. But the animals have to have the ability to selectively ingest it because a lot of it's not very useful. And then they have to have the ability to digest it after they've selected the highest quality material. So those are the two essential requirements to being a successful consumer of biofilm.
And I was interested in the pairing of the feeding and the non-feeding lampreys. What sort of hypotheses do you have about why they're paired?
I don't have a sophisticated hypothesis myself. The thing that comes to mind is that when the prey of the chestnut lamprey are abundant, it has the advantage. But when they're not, then the non-feeding brook lamprey has the advantage. And the prey populations are more likely to wax and wane over time. So Are you familiar with the paradox of the plankton? Do you know that story? So, one of the founding fathers of ecology, G. Evelyn Hutchinson, who wrote a lot of seminal papers in the 40s, 50s, and 60s. He came up with the idea of the n-dimensional niche, which you might have run across in a textbook. But he described this thing called the paradox of the plankton because there are hundreds and hundreds of species of algae that make up the plankton in the ocean, and yet it's constantly mixed, and it looks like a homogeneous environment. So why doesn't one of them outcompete all the rest? Is the paradox. And the answer is it's never an equilibrium situation. There are always things that are changing. And so that would apply to the pair of lamprey, that they are almost certainly not in an equilibrium static environment. The abundance of spawning habitats, the quality of the biofilm might vary from year to year. It's interesting that lamprey larvae do really well downstream from sewage treatment plants because that waste really stimulates the production of biofilm. So there are local variations. That's about that.
So, service that is poisoning the larvae, they know where the 1A habitats are, they specifically target those as close as possible?
Actually, they can't do that because they're too local, and chemical goes everywhere in the stream. So what they do is they survey the whole stream, but they target the 1A habitat because they expect to get the most results there. And then based on what they learn, they decide when they're going to poison the stream. But you have this poisoned mass of water that will flow for many, many miles, essentially down to the mouth. And it doesn't break down quickly, so it continues to be active and you can't focus it. Actually, there's an exception to that. In addition to lampreys spawning in streams, there are some areas in large rivers, particularly the St. Mary's River, where the lamprey spawn and the larvae live in deep parts of the river, tens of meters deep. And these are big rivers, so they're not places where you can use this soluble poison to get to the larvae. So instead, they've produced a granular form of the lampricide. Basically, it's just coated on the sand particles, and then it's deposited in an area where divers have found the larvae. You can do spot treatments that way. They're not as effective as using the dissolved chemical. They work to some degree.
Any other questions? No? If not, thank you very much.